Hello, everybody, and thank you very much for joining us for the third installment of the Spring 2020 Closer Look Journal Club. I'm joined here today by my colleagues Manuel Rausch, Matthew Bercy, Johannes Weikenmeyer, and Adrian Buganza-Topole. And today we're really excited to host Michael Albro. Michael completed his PhD at Columbia University in biomedical engineering. He was then a postdoc at Columbia and a Marie Curie International Fellow at Imperial College London before starting his lab at Boston University in 2017. He does really interesting work in both the biomechanics and mechanobiology of musculoskeletal tissue. He has multiple really exciting projects going on, and today we're going to hear about just one of them, specifically the Raman needle arthroscopy for in vivo molecular assessment of cartilage project. And I'm really looking forward to hearing more details about this project. I've been hearing sort of bits and pieces about it over the past couple of years, and the suspense has been building. So Michael, please go ahead.
I hope I don't disappoint. Yeah, no, thank you everyone for inviting me. I'm just thrilled to be here and to be able to share this project. As the organizer specified, I think they want to focus for the most part around this paper that we recently published in the Journal of Orthopaedic Research. And I decided to kind of structure this talk around a little bit about the background and motivation of how we got to this platform and conceptualized it, and then some of the work that we've done since this publication as the project continues to grow. But first I just want to kind of really acknowledge my collaborators on this project. This is a really, I'd say, somewhat intense collaborative effort. There are a lot of moving parts to this project. So Mads Burkholt's a professor at King's College London. He's an expert in Raman instrumentation. Brian Snyder's a orthopedic surgeon, researcher at Beth Israel. Mark Grindstaff is a chemist at Boston University. And then Thomas Shear is a veterinary surgeon at the New Bolton Large Animal Center. And really, like, for this to move forward, it requires all these moving parts. So, I would very much think of this as a very intense collaborative effort. But starting off, the project really starts with osteoarthritis and articular cartilage. And everyone's familiar with this tissue, this soft connective tissue that lines the end of long bones. It's smooth, gives rise to really nice, low friction, great tribological properties that allows for smooth, pain-free locomotion. In osteoarthritis, the tissue begins to degenerate. And over time, eventually you'll be left with bone-on-bone contact, which can be incredibly painful and debilitating. And I think, as many people know people who have osteoarthritis, so it's well known that currently there's no approved FDA therapy, drug or other therapy, that can mitigate the progression of this disease or reverse the progression of the disease. And therefore, for the most part, this is treated with pain management up until you reach the age where it's decided that you should have a joint replacement surgery. And I think to understand really the challenges in terms of therapy development, we have to understand about the biomechanics and the composition of cartilage. And this is a soft connective tissue that is predominantly extracellular matrix, ECM. The articular chondrocytes make up just 1% of the tissue volume. The rest is matrix. And the matrix is optimized for its mechanical performance, where you have these dense, large proteoglycan aggregates that are immersed within a type 2 collagen fibril network. The GAG, or proteoglycans, are able to resist compressive strains. The collagen resists tensile strains. The tissue is 90% water, and the highly charged GAG basically has this water attracting quality to it, which allows the tissue to strongly pressurize upon loading. And that pressurization gives exceptional resistance to compression and a low friction interface. So in a sense, all these agents working together give rise to the remarkable load-bearing and tribological properties of this tissue. In osteoarthritis, the composition starts to degrade. And these histological orsigrades kind of show the stages of how that transpires. These are slides that are stained. The red is the proteoglycans. The blue is the counter stain that shows the collagen. And initial stages of OA degeneration consist predominantly of GAG depletion, that enzymes start to break down the proteoglycans, they get depleted from the tissue. There's some collagen breakdown, which causes some swelling, but overall, early stage OA is associated with the tissue getting softer, the friction coefficient increasing. So all in all, what you get are these very undesirable positive feedback loops where initial degeneration promotes further degeneration. And the further degeneration consists of the collagen network just physically eroding and breaking down over time until you're eventually left with bone-on-bone contact. And the therapies that are currently being developed in the clinic are inspired in a way of trying to either slow the progression, so either stop going from 1 to 2 or 2 to 3, or potentially reverse that degeneration. So if you are able to find someone with grade 2, reverse it and go back to grade 1. And for the most part, what that means are therapies that are aimed to either stop the GAG loss or replenish the GAG loss, the lost GAG, right? So these are things like, enzyme inhibitors that will stop the GAG from being cut up, anabolic growth factors that can increase the biosynthesis of chondrocytes for more GAG. So you're trying to restore the composition, the GAG content, and restore the stiffness, the mechanical properties of the tissue, right? But the challenge about bringing these therapies into the clinic is that it's very difficult to see if they're working or not. And that's because the diagnostics is not keeping up with the biology. And currently, the diagnostic platforms for seeing if someone has arthritis consists predominantly of radiograms. So basically, if it's this image on the right, and what it's looking at is the gap in between the bones. What's the gap? It's the cartilage. So you're looking at the thickness of the cartilage, and over time, the cartilage will erode, that space gets thinner and thinner. But in the end, this is really biased to late stage disease pathology, where the collagen at that point is just ripping apart, and there's not much you can do in terms of a therapeutic platform at that stage. MRI will give you better assessments, but MRI is really nice, I think, for looking at bone cysts, inflammation, but in terms of assessments of the cartilage composition or the mechanical properties of the tissue, MRI is really not geared for that. So the consequences of this is that it's very difficult to see if a therapy is restoring the composition and restoring the mechanical properties of a patient's cartilage, right? So currently, clinical trials are very much rely on patient-reported outcomes. So ask the patient basically, how much pain are you in? Give us a number, on a Womack scale from 1 to 10, and then give the therapy, and a year later, ask again, how much pain are you? So in the end of the day, this is really challenging, especially because these therapies are not geared towards reducing pain per se. They're geared towards replenishing the composition of the cartilage. So what we're interested in is expanded characterizations or expanding technologies for diagnostics. And we're looking in this technology of Raman Spectroscopy to see if it can assess cartilage health. Raman is an inelastic light scattering technique. So what this means is when you hit a specimen with incident photons, high intensity laser light, basically a portion of the photons will interact with the molecular bonds of the tissue specimen, and the vibrational modes of those bonds can induce a small slight shift in the wavelength of the photons. And if you could measure those shifted photons, you can interrogate information about the molecular structure of your specimen. So in the end of the day, what this really means is you can measure this spectra that represents more or less counts of the different molecular bonds or bond fragments inside a tissue, like the phosphate groups, the sulfate groups, carboxylic acids, amides, things of that nature. If you have purified chemicals and you take Raman acquisitions, you get these really nice Raman molecular fingerprints. So like here, there's the fingerprint of hydroxyapatite, a characteristic lipid, proteins. So, this is a really good technology for identifying like unknown molecules, unknown chemicals. And because of that, there are a lot of really cool, I think, applications that have been investigated with Raman. I think one of the cooler ones is in the field of art forgery, where basically this is really good for looking at the pigment in a painting and trying to determine if the the alleged masterpiece, the alleged Rembrandt, was actually made in the 17th century in the Netherlands or not. So there are definitely applications where this is really, I think, straightforward to implement. And in recent years, there's been a lot of interest in trying to evolve this technology into biomedical fields. And that's kind of the wave, I think, that we're riding a bit. And we're trying to answer this question of, can Raman spectroscopy be used as a clinical platform to assess cartilage composition and material properties? So I'll quickly talk a little bit about how I got interested in this topic in general. Or actually, no, sorry, I forgot about this slide. Just giving you a little bit of the conceptual breakdown here is how this would be used in the clinic, is where you would need to take the Raman instrumentation and miniaturize it into a Raman arthroscope that can be inserted either through a needle cannula or an arthroscopic portal. You would have a lens at the end, and you could tap a patient's articular cartilage and assess this optical biopsy for measuring composition and functional mechanical properties. The innovations that would have to be employed is, one, instrumentation. You would need to miniaturize this and make a probe that can be used in the clinic. And then two, the spectral analysis. You would need to be able to extract meaningful clinical biomarkers that would be informative about a patient's cartilage health or disease state. And for a purified molecule, that's more straightforward. But when you have a tissue specimen that's made up of tens of thousands of proteins and sugars and lipids, this becomes a significant challenge in terms of how do you interrogate that information from this very complex composite Raman spectra. But more or less, I think the workflow that we're interested in is taking a Raman spectra of a cartilage specimen and breaking it down into the underlying component spectra of the extracellular matrix, and then using that information to try to get predictions or measures of the functional mechanical properties of the tissue. So now I'll talk a little bit about my background and why we became interested in this. And this really started, I think it was 2015, where me and Mads Berkholt, he's now at King's College London as a PI, but we were both at the time postdocs at the Stevens Group at Imperial College. Mads had worked extensively on Raman instrumentation for cancer diagnostics, and I was very much interested in the cartilage ECM. So we started this project, which was, I think, less ambitious in nature, of just seeing, can we take cartilage histological sections and use Raman to quantify the distribution of cartilage extracellular matrix, right? So histology, as everyone knows, is a very qualitative field where people are looking and describing distributions of different stains. So we wanted to try to bring some quantitative rigor to this field. So we would put our sections under a confocal Raman microscope, and we performed a technique called, some people call it Raman spectroscopic imaging. It's also referred as hyperspectral Raman imaging. But you basically take a Raman spectra at discrete points in the tissue, different pixel positions, and then you generate this very large hyperspectral Raman data set, where at every pixel you have a spectra of the tissue. And then you could create an image in whatever fashion you want. If you're interested in phosphates, you could look at a phosphate peak and plot that over the sample to get this really nice image of phosphate in your tissue sample. What we were interested in is the main ECM components of cartilage, the GAG, the collagen, and the water. So we employed this technique called multivariate curve resolution, where we would break down the tissue spectra using this linear model consisting of these essentially weighted contributions of the GAG and the collagen and the water contribution to the tissue spectra. And what MCR is able to do is it's able to curve fit for the GAG spectra that represent the overall GAG throughout your hyperspectral data set, the collagen spectra and the water spectra throughout your entire data set, and then these weighted regression coefficients at every single pixel, right? So it's a lot of information you're extracting with the curve fit, but it works well if you have a very large data set. And we did. Each image was on the order of about hundreds of thousands of pixels. So you're able to apply this, and it works quite nicely. You're able to, this is the cartilage, the representative cartilage spectra, and these are the extracted GAG spectra from that cartilage spectra and the collagen spectra. And if you compare that to the spectra of just a purified chemical that's thrown under the microscope, purified GAG, purified collagen, you see really nice agreements, suggesting that the technique is working. And if it does work, now you have these regression coefficients. You have essentially the relative marker of the concentration at every pixel in the tissue. So we were able to generate these really nice maps, quantitative maps, to see how the GAG and the collagen and the water would vary through the depth of native articular cartilage. So GAG and collagen increases through the depth, water decreases through the depth. This is well known, but now there's a quantitative numbers that can be applied for that variation at very high spatial resolutions. You could also do this with engineered cartilage as well, where you see these heterogeneities that develop, based on just the growth profiles of how the tissue forms. And you could kind of look in different length scales where you can get these very high resolution images, that are submicron in resolution. You could also look at how the distribution of GAG and collagen through the tissue. So, that was our prior work in kind of another lifetime. But we were definitely emboldened by these results, and we decided next, could we really apply this to probably the grander challenge of clinical diagnostics? So, at the time, I think this was around 2017, 2018, we both had our independent research positions starting out, me at BU, Mads at King's, and we decided to go for it. We combined all our extra research dollars that we could find, and we decided to design and fabricate this $20,000 Raman arthroscopic probe that consists of just filters. There's a laser light that is aligned through this long, hollow needle tip probe here. There's a very small 2 millimeter ball lens at the tip as well. And basically, you just touch down to the cartilage and you get this Raman optical biopsy of the tissue. The light is backscattered and goes through a fiber coupled spectrometer. So this is the instrumentation that was developed. And the next question is the the analysis of the spectra and how do we employ that? So, in the past, as I just showed, we use this technique of MCR, but it's really not feasible for tissue diagnostics because you just don't have nearly enough data. It would be very time consuming to biopsy 1 million pieces of cartilage one by one in a handheld configuration. So what we did was, instead of using MCR, we plugged in reference chemicals, spectra from reference chemicals. So we plugged in known spectra of GAG, collagen, and water that were acquired with the probe, and then put it into this linear regression model, where now we're only curve fitting for the regression coefficients, which we refer to as Raman biomarker scores. So for every tissue specimen, you're getting a GAG score, a collagen score, and a water score that roughly relates its contribution to the tissue spectrum. And by doing this, you're able to recreate this really nice 2D stacked area plots that show the cumulative contribution of each component to the tissue Raman spectrum. All right. So you're able to now interrogate some biochemical information from the Raman spectra that relates to how we think in terms of cartilage composition. So over the next few months, years, we've been interested in performing ex vivo model systems to basically see how well this platform might work, right? Doing this in the clinic is obviously very challenging. So a lot of our work has been developing these proof of concept models to see the efficacy of the platform. So one of the things that we were most concerned with is, can this device detect GAG depletion, which is associated with early osteoarthritis, right? And this is, as you can see in these ORSI grades here, one of the things that happen earliest in disease progression is that the GAG is depleted from the surface. So can you quantify the GAG, and can you quantify the softening that happens as well during early osteoarthritis? So, our model consists of bovine cartilage explants that were procured from femoral condyles of cows. And then you could deplete them in a really nice and predictable manner where you treat them with either guanidine or hyaluronidase. Guanidine will give you this nice kind of slow depletion of GAG, and hyaluronidase basically just cuts it all out of the tissue, if you will. So you get these really nice stepwise depletion model of GAG from these cartilage explants as occurs in early stages of osteoarthritis. And then you can compare Raman measurements to direct biochemical measurements and mechanical property measurements. And this is showing the Raman spectra for these different GAG depleted cartilage specimens. And you see, some nice predictable peak decreases at some GAG associated regions. And then we're able to employ our multivariate regression model here. And what you see is that you're extracting col scores and water scores, which really don't change that much as expected. The collagen content and water content of these tissues is really not changing that much. But the GAG scores show this really nice stepwise decrease as we deplete more and more GAG. And again, showing these 2D stacked area plots, you see very nicely that the GAG contribution that makes up a sizable portion of the healthy spectra is now lost in the degenerated samples. And when we compare the GAG scores, the GAG biomarker scores to direct measures, you see that it very nicely correlates with the assay measured GAG content in the samples, and it very nicely correlates with the mechanical properties of the samples. So what this means is just with a purely light-based optical biopsy, you can get these very nice assessments of the composition and the mechanical properties of the tissue, right? Other things that are kind of cool is that you have the ability to look at different spatial aspects of the tissue. And with OA, GAG depletion does not occur uniformly. It occurs typically from the surface down as the enzymes are more concentrated in the synovial fluid. So you could create a model where you digest the tissue with trypsin, which creates this depth-dependent depletion of GAG. And then you can measure the Raman spectra with different lenses as well. And we've used this ball lens, which has about 170 micron depth of penetration, or you could change the lens geometry to make it a convex lens, which will go interrogate deeper into the tissue. And what you see here is that if you use the deep penetrating lens, the correlation with the GAG content as assessed by the histological stain is not all that impressive. And because you're picking up too much deep zone information when a lot of the interesting degeneration happens at the surface. But if you use the ball lens, you get a much better correlation with the GAG content of the tissue. And you're also able to get this really nice correlation with the elastic modulus as assessed at the surface of the tissue as well. So you can play these games by using different lenses if you want to get information from different regions of the tissue. The other thing you can do is look at structure of the tissue as well. And you have the ability to look at the thickness of the cartilage by analyzing the signal from the subchondral bone. So this shows basically cartilage specimens, bovine specimens that were just sectioned to make the chondral layer different thicknesses as a very rough model. And then you could take the Raman spectra of each specimen. And what you see here is that if the chondral layer, the cartilage layer is very thin, you pick up a lot of bone signal because you're very close to the bone. And as the cartilage gets thicker and thicker, the bone signal starts to diminish. So, you could add bone in as another term into the multivariate regression model. And then you get this bone score, which correlates very nicely with cartilage thickness, which I think shows you have the ability to look at composition, but also structure in just one quick acquisition. The other thing we were interested in is these models are really coarse. You're basically just, changing one thing in a very controlled manner. And we were curious as to how these biomarkers might change in a real tissue system where you're going to have variability of not just, one parameter, but all parameters simultaneously. So you can take these osteochondral explants from a sheep's knee, and you actually see a lot of variability just based on the anatomical position where, if it's on the tibial plateau or the condyle, they're going to have different properties. They're going to have different GAG contents, water contents. The thickness can vary from 200 microns to 1200 microns. The modulus can vary by a factor of 5. And we were wondering, can the Raman really pick up this anatomical variability? And when we perform measurements, you see quite nicely that the Raman gag score correlates well with the variability in the gag content. The water score correlates well with the water content, and the thickness can be predicted by that bone score as well. I think the other thing that's cool about this is the ability to use these biomarkers to predict or measure the mechanical properties of the tissue. So in the previous model I showed, we were just using the gag score to predict the mechanical properties of the cartilage. And if you do this with this ovine model, I mean, that does a pretty good job of it, because the gag really is the dominant factor in predicting the compressive stiffness of the tissue. But you have all this additional information like a water score, bone score, collagen score, and you can even interrogate more information from the spectra if you wanted to. So, you know, it provides this really unique opportunity to use all these biomarkers in concert to get better predictions of tissue function. And this shows nicely that if you develop a model based on this linear combination of the gag score and the water score, you can actually get greater predictive power in predicting the elastic modulus of the tissue. So I think it really shows, you know, the power of this technique and using just multivariate approaches to predict, you know, things about tissue functionality. This was the next step of the publication, where we actually did this in vivo, and this was performed at the New Bolton Large Animal Facility associated with the University of Pennsylvania by Thomas Shear and Brian Snyder. And this is us using the probe through a mini arthrotomy and also using it through an arthroscopic portal. And it shows quite nicely our probe has the ability to be used in, you know, a clinical setting, can be used in vivo, and you can, you know, monitor it as you are in contact with cartilage and potentially perform, you know, site-specific diagnostics. And this is the spectra, the process spectra that we get out of these in vivo experiments. And they're, you know, very similar to the quality and composition of the spectra that we see when doing this in vivo or ex vivo as well, suggesting as a nice proof of concept that this platform really does have the potential to be used clinically. So, you know, that is the work that was published in this paper, and I think it shows quite nicely through a host of model systems that the probe can measure these ECM-specific biomarkers for assessments of cartilage composition and material properties, and we also showed nicely the feasibility of doing this in vivo. I'll show some of the work as well that we've done since then, which I think is really exciting, where we're interested much more in diagnostics of naturally degenerating cartilage. So we've been employing a number of different model systems of using different species. And one of them is bovine cartilage that we find in different degenerative states. You know, degeneration can be, to get a rough quantification of what do we consider degenerated or not, you know, people in orthopedics generally use these clinical outer bridge grades, which are, you know, coarse. You get a grade from 0 to 4, which indicates different levels of degeneration. So it's a nice starting point, I think, to at least classify the different tissues you're looking at. But what you're able to see here is these are condyles from bovine joints. On the left, these are healthy condyles. They're outer bridge 0. They are, you know, beautiful, pristine. And on the right, these are just from a different animal, and it has a chondral lesion. There's a focal defect, as you can see in the circle region. It's subtle, but that is an outer bridge 2 defect, a small chondral defect in the cow specimen here. The surrounding cartilage is macroscopically healthy. You don't see any sign of degeneration there, so it's an outer bridge 0. And what you now have the opportunity to do is, you know, look at the ability of Raman to potentially predict changes in composition and properties throughout these different regions of these joints. And so if anyone's ever wondered, do cows get osteoarthritis? The answer is yes, they most definitely do. And you could actually see a lot more degeneration than this in older specimens. So our workflow is breaking this up into osteochondral blocks, doing analysis with mechanical testing, Raman diagnostics, biochemical assay measures. This is the indentation testing we do for the elastic modulus of our tissue specimens. And when you look at the results, I find these are very recent results for us, and I find them fascinating, because what's really cool about this is if you look at the GAG content, this is in 4 healthy joints, which is outer bridge 0 everywhere. And then this is 1 joint with a lesion. But we did the analysis in the outer bridge 2 lesion site, but also the outer bridge 0 peripheral tissue. And you can see that both tissues are severely GAG depleted. So even though the outer bridge 0 here looks healthy, visually it looks great, it's depleted of GAG. And because it's GAG depleted, it's much softer, right? So a clinician would never notice that, never be able to pick that up, but that tissue has undergone degeneration and maybe at this point is vulnerable for, rapid erosion, right? So it's something that you cannot pick up visually, but the Raman probe is able to see that. The Raman GAG score is able to show significantly reduced GAG contribution in this clean region of the tissue, as well as the defect site. And when you take all this data and you throw it together, you get this really nice correlation with the GAG content, as we saw before, as well as correlation with the elastic modulus of the tissue. So again, this is really showing that Raman has the potential to diagnose tissue compositional changes that the eye cannot see, which I find exciting. We can also do this with human cartilage, and this is a 32-year-old healthy knee. This is, for 32-year-old, this is wonderful, pristine, beautiful cartilage. You don't see this very often. And then this is the cartilage for a 65-year-old, which doesn't have to be osteoarthritic, but in this case is, where in this medial condyle, you have an outer bridge score 4 with the subchondral bone exposed. The lateral condyle looks pretty good, except for this slight blistering here. Most of the surface of that tissue is in a pretty good state with outer bridge 0, no degeneration. And you have the ability to now diagnose these different lobes of the condyles of these specimens. And you see that in the OA knee, you have significant softening in the outer bridge 4 condyle, but also in the lateral outer bridge 0 condyle. So once again, you have a lot of surface softening that cannot be visually seen. And then again, the Raman GAG score is able to pick that up and show that both tissues have this more or less the same mechanical properties, even though one looks great and one doesn't, right? And again, correlations with mechanical properties overall look pretty nice, okay? So the last thing I want to just quickly talk about is our latest work, which is looking at the ability of this technology to look at chondroregenerative therapies as well. So while there are no drug-based treatments to reverse the course of the disease, there are surgical treatments to try to regenerate cartilage in patients that are currently employed. And for the most part, these are used in a subset of OA, in a condition called PTOA, post-traumatic osteoarthritis, which is degeneration that sets in after a traumatic joint injury, like an ACL tear or a meniscal tear, or shortly after you will get a chondral lesion or could get a chondral lesion showing up on your cartilage surface. And that will probably quickly lead to osteoarthritis. And because this usually occurs in younger patient populations, surgeons generally try to treat this pretty aggressively. And there are a host of chondroregenerative treatments that are employed, like they'll inject cells in the defect site. They'll try to infiltrate the site with bone marrow to promote healing. Cartilage tissue engineering is an exciting platform that's also used to repair chondral lesions in PTOA. But again, the challenge with this is that none of these therapies are providing the kind of clinical outcomes that one would hope for in terms of surviving decades and not just years. And it's very difficult to assess the efficacy of a platform in a patient because all you have is the ability to do MRI or a radiograph at this stage. So it's difficult to see if these regenerative platforms are really recapitulating the composition and functional mechanical properties that one would hope for. So our work is kind of focusing on this path of cartilage tissue engineering and Raman diagnostics of cartilage tissue engineering. And just briefly, the platform is one where you take chondrogenic cells, those are either patient chondrocytes or MSCs, you passage them, you see them in a hydrogel scaffold in 3 dimensions, and then you implant that cell scaffold construct in the defect site and hope that it will lead to cartilage growth and repair. This is an ex vivo examination. It shows kind of in some sense how well the platform works, where going from day 0, which is just a hydrogel scaffold, day 45, you can get a tissue that looks and feels just like native articular cartilage. And if you give the proper growth factor cocktail, you can potentially recapitulate the true functional mechanical properties of native cartilage. So in many ways, this field has come very far, but again, it still has ways to go in the sense that the tissue does not survive as long as one would hope. So there's a lot of work in manipulating cell sources, scaffold chemistry, and growth factors to try to improve clinical outcomes. So our question on this front was, can our Raman platform perform an optical biopsy of cartilage repair tissue to quantify the composition and functional mechanical properties as the tissue is regenerated, right? And this is the Raman spectra of an engineered cartilage construct at a given time point. So this is really recent work for us, but what we did was this proof of concept study where we were able to grow engineered cartilage tissues in the lab and then take our probe and see if we can predict and measure its composition and mechanical properties. The challenge here that we encountered and realized really quickly is that when you try to do this, you don't just have the complications of different ECM constituents which overlap, where you have collagen, GAG, and water, but now you have the additional scaffold constituent as well, which is there at very high concentrations and really clouds your signal. So like basically, if you're using agarose, hyaluronan, you've created this huge overlapping mess. And the question really for us was, can our multivariate models tease out the contribution of all these different constituents in the engineering repair tissue? So the experiment was basically taking chondrocytes, seeding them in an agarose hydrogel scaffold, cultivating them for 56 days with or without TGF-beta, which could give us different growth profiles, and then performing endpoint analyses where we would do mechanical testing, Raman analysis, and biochemical assays of the ex vivo repair tissue. And when we first looked at the data, this is essentially the raw data, if you will, of a tissue at day 0 versus a tissue at day 56. And I'll admit that looking at this, when the student first brought me this, I was horrified by this because it looks like a mess. I was, you're expecting some nice peak that would gradually increase as the tissue is growing, and it's like, it just seems like a lot of overlapping mess, if you will. And if you look at the tissue specimens, I was wondering if I might be able to assess growth better by eye than with this very expensive, highly precise instrumentation. But what's cool about it is when you apply the multivariate regression models and you add in the scaffold as an additional term here, you're able to make a lot more sense of the data. And you can see here that while the spectrum might look very similar, at day 0, it's really dominated by the agarose scaffold and the water. And at day 56, it starts to become dominated by the GAG and the collagen that's deposited by the cells. So you can look at the evolution with time, and at day 0, as you move forward, you get more deposition of GAG and collagen with time. When you add the anabolic growth factor, the deposition of GAG and collagen occurs much more rapidly. You can look at the biomarker scores quantitatively, and you see here that the GAG scores increase with time, the collagen scores increase with time. They increase further with the anabolic growth factor. The water scores decrease with time, and the scaffold decreases with time, which makes sense. You are, as the tissue is growing, you're replacing the water and the scaffold with the extracellular matrix, the GAG and the collagen. So this kind of passes that reality check, if you will. And then you have the ability to look at this quantitatively and see, does it agree with direct biochemical measures and mechanical assessments? And you see that it does very nicely. The Raman GAG score is highly predictive of the GAG content in the tissue and predictive of the mechanical properties, the equilibrium Young's modulus of the tissue. You also have the ability to monitor the water content as well, which agrees quite nicely with direct water content measures. So yeah, I mean, that's kind of, I think our latest data in a lot of ways, but we're also interested in many different expanding orthopedic applications. We have a grant from the Musculoskeletal Transplant Foundation to look at the quality of osteochondral allografts before they're implanted in patients. And that's not an in vivo platform, that's a purely ex vivo platform that has a lot of clinical utility. We're also interested in if Raman can be used for, different musculoskeletal connective tissues. And this is, our probe with an intervertebral disc. And you can see, for those who know disc anatomy, the nucleus propulsus, which is this inner region, is rich in GAG and water, and that shows up nicely with the Raman spectra. And the annulus fibrosus, which is a fibrocartilage, type 1 fibrocartilage network, has much less GAG, and you can kind of see that quite nicely. We're also developing what we're calling a Raman plate reader for ex vivo analysis. So you have the potential to put live tissue specimens in 96-well plates and just with a probe monitor them over time. It can be done in the incubator. It can be done by taking the plate out of the incubator, but it gives you the potential to do this really nice high frequency repeated measure analysis on live tissue explants. And this is just proof of concept, us showing that, I think this is very high temporal resolution, twice per day, looking at the decrease in the GAG score as live cartilage explants are being treated with a cytokine, interleukin-1 alpha. So yeah, I mean, that's where we are with this. And just in conclusion, I think, a lot of our models are showing nicely that the probe is able to extract these nice ECM biomarkers that are predictive of composition and material properties. It can be used for native cartilage to look at OA, degeneration, or potential regeneration with a treatment. It also seems to be quite adept at measuring the composition and material properties of regenerating cartilage. We're one of the few groups to show that Raman can be used in vivo for assessments of composition of tissues, which I think is a really nice proof of concept for moving forward. And then where we see this going in terms of clinical potential, I think very soon this can be interfaced with clinical trials. And I think that would be a really exciting approach to be able to look at the efficacy of some of these emerging therapies in improving the GAG content, the composition, and material properties of patients with OA. And then long-term, once the therapies develop and are brought into the clinic, now you would have this platform that can be used in parallel with treatment courses, being able to diagnose osteoarthritis at early stages of disease progression. You could potentially do screening for patients who are at high risk of developing OA, such as patients who have a traumatic joint injury. And then it can be used to monitor how well those treatments are working with individual patients. So I see a lot of different areas that this could go into, and in many ways we're at the very early stages of the work, but I think we're pretty excited by it. So yeah, just want to acknowledge everyone who's worked on this study. Our funding sources: the Arthritis Foundation, MTF Biologics, the Boston University Center for Multiscale and Translational Mechanobiology, our lab, the lab at King's College, Beth Israel, Grindstaff's lab, and then the New Bolton Center. One thing I want to highlight is on the Journal of Orthopedic Research paper, we definitely had humble beginnings when starting off this project, and that study has three lead co-authors, three lead authors who are all undergraduates here at Boston University: Kimberly Krupa, Maggie Wu, and Zhengchen Zhang, who are, Kim is now at Columbia pursuing her PhD, Maggie's at Michigan pursuing her PhD, and Andy is applying now to PhD programs. But I think it really shows, I mean, the students did tremendous work on the initial stages of this project, and I think it's a real testament to Boston University, the quality of students that are being generated at the undergraduate level. But yeah, that's all I have. Thank you all, and happy to take questions.
That was awesome, Michael. Thanks so much. Um, since we only have like 12 minutes or so, let's jump right into questions. Spencer, if you want to go ahead.
Yeah, thanks, Michael. Really nice study. I had a question about, you talk about GAGs and collagen as categories, but of course there are multiple types of collagen, multiple different types of GAG. And I assume for something like cartilage, when you're setting up your standards, you could do, call2 and aggregant or something as predominant, compositional components. But what about more generally for other tissues where there might be a mix of GAGs or a mix of different types of collagens? Would you have to have standards that just match in that way, or how do you handle, variations in those standards?
Yeah, great question. So it really depends on the tissue of interest. And in many ways, you hit the nail on the head there that cartilage, the intervertebral disc, due to, I think, the simplicity of the ECM structure, these are really nice tissues for Raman diagnostics. It's a low-hanging fruit in some regards, and there are other tissues that are going to be more complex. I mean, certainly any tissues that are vascularized, like bone, is just more of a challenge. But I would say, for connective tissues which are avascular, I think they all provide this very unique opportunity for basically getting clean isolation of the extracellular matrix. If you're interested in digging more into greater specificity with types of collagen, with types of GAG, it can be done, but it's more of a challenge. And at the end of the day, you need a model that's going to allow you to confidently extract that information, right? So I think in terms of collagen subtypes, that's something we are looking at, but in the end of the day, it's difficult to get large amounts of different collagen subtypes that are purified enough to do the analysis. So I think the answer is yes, you're asking the exact right question, and that's the right direction to go into, but it is more challenging than what we've done so far.
Great, thanks.
Are there any other questions from the audience?
I think my question was a little similar to Spencer's, but maybe a little more concretely focused on, of course, what I'm interested in. So generally speaking, this should be generally applicable to all kinds of tissues, right? In a sense that you're really just looking at sort of the vibrational modes of chemical bonds. And I think in a lot of ways, all degenerative diseases have sort of similar origins, or at least sort of the similar characteristic in that, your normal composition and your normal structure kind of degenerates, and then the tissue loses its function and fails in whatever capacity. So of course, now I'm sort of imagining how this could be applied to other tissues. What is the, how dynamic can the tissue be that you're imaging, right? So I'm thinking, for example, heart valves. Like how much acquisition time do you need? And then can you imagine that this could be similarly done on tissues, that are hardly collagenous, but maybe have a small GAG component, et cetera, et cetera. So I'd just be interested to hear your thoughts. Can we look at valve degenerative disease, for example?
Right. I think so. I think it's fair to say that every tissue in the body is Raman applications for every tissue is being investigated by someone right now. I think is fair to say, and different tissues will be easier than others. So one thing is I think we have to break down Raman into two categories. One is imaging, which is using a microscope to either look at a dead tissue section, or you could do live cell imaging with Raman. And there you have the ability to get, really nice high resolution images, but of course it's all ex vivo. Or you could use probe technology in vivo, but it's more, point by point analysis. In terms of tissue composition, it depends on what you want to extract and what your ultimate end game is. So, people have been doing Raman for cancer diagnostics for a long time, and they don't extract information about each molecular constituent just because the cell changes are just too complex. And they apply PLS models to basically be able to just get a diagnostic. Is this cancerous? Is this, benign, or is it cancerous, right? And for, clinical workflow there, that's enough, and that's incredibly valuable in its own right. For cartilage, I think our thought is it's not enough to say, is it OA or is it not OA? You need to know, like, why is it OA? Why is it degenerating? Because that's really the way to get a robust assessment of what's happening. So I think there are definitely, those are two ends of the spectrum. And every tissue is kind of, I think, going to fall a little bit more to one end versus the other. So a heart valve is a great example of one where you might be able to get enough information about the composition and extract that information in a meaningful way. You could also do tricks like polarized Ramans. You could get assessments of the organization of the collagen as well. But I think, you have to really start with understanding what is the composition of the tissue and what are the salient features that you really want to diagnose to get this into the clinic.
Thank you, Michael. Spencer said already, and I think Emma does fantastic work. And I really love the linear progression of where you started and where you took it. So I think it's beautiful.
Thanks so much.
I see it. Jill raised her hand. Jill, please go ahead.
Hey, Michael.
Hey, good to see you.
Hey. I guess I was wondering, with your optic probe, do you have the ability to change the penetration depth on it? To get depth dependent gag properties or collagen properties?
Yeah, so there are definitely different techniques. There's a technique called SOARS, which is spatial offset Raman that people are looking at, where you could potentially multiplex and get depth position simultaneously. It's challenging to implement for sure. But our approach of this has been to just modify the lens that we put at the pro tip. So I think I showed one slide where there are just two lenses, and one you get more surface acquisitions and one you get deeper acquisitions. But you could do that in a very, I think, with micro machining, you can make lens of any geometry you want within reason, and have the potential to generate really tight control over where in the depth you're interrogating.
Cool. And then I kind of had one other question related to your relationship between your biochemical assays and your coefficients of your spectra. They weren't always linearly correlated. So do you have a reason for why that occurred? Or I guess I would have expected them to be always linearly correlated, because that's what I assume with FTIR, which is different spectroscopy, but I would have thought the same principles applied.
I can't get through a talk without getting asked that question. The answer is, one, keep in mind that, the spectra, I didn't go through the algorithm of the pre-processing, but it's always normalized to the area under the curve. So basically the signal is always the same, in terms of the area of the spectra. And then the regression coefficients, they're not quantifications of the amount of a chemical in the specimen. They're not absolute quantifications, right? They're biomarkers that reflect the contribution of a molecule to the tissue spectrum, right? So there's nothing fundamentally that says that they should linearly correlate with concentration. They should be proportional, is what's there. And I think, to answer the question, when you're looking at the native tissue results, that is a model with only three terms in it, right? So you have, two degrees of freedom essentially, because it all has to add up to the area under the curve. But when you look at the nonlinear work, that's in the tissue constructs, where we have the additional scaffold term as well. So basically these things have to fluctuate in proportion with each other. So, one of the things you'll see there is actually the agarose score is decreasing with growth. The agarose is not disappearing with growth. The agarose is stable, and it's in there at the same concentration for all 56 days. But the contribution of the agarose to the Raman spectra decreases as the GAG and collagen contribution increases. So, I think that's really why this happens. There's nothing fundamentally that says they have to be proportional. They have to be linearly proportional.
Sure. So the total concentration of agarose was not changing, but the relative concentration of agarose
relative contribution was changing. Exactly.
Yeah. Great. Thanks.
Yep. Thank you.
Yeah. Yeah, I think we are basically out of time right here. So I just want to thank Michael one more time for such a fantastic talk. It was really nice to see what went into this paper and then also to see how it kind of ties into your lab vision. So yeah, thank you, Michael. I think I speak on behalf of everyone, and I will go ahead and stop the recording.
Thank you all for inviting me. This was such a pleasure.